Hi, so this is part 2 of chapter 4, parts of a eukaryotic cell. So the first thing I have here is for you to write down what is the definition of a eukaryotic cell. So what I have on the PowerPoint slide here is what the eukaryotic cell, basically the definition, right? So there's four major components that a eukaryotic cell needs to fulfill. The first one and the second one are related, and those are the most important. So eukaryotic cells have membrane-bound compartments. We will talk what a membrane is, but we talked about this a little bit. You know, it's a phospholipid bilayer in chapter 3. So this yellow portion is the outermost membrane. We call this the cell membrane or the plasma membrane. But if you look inside, we have this guy here, we have this structure in pink and red, we have this blue-purple structure, we have this guy here. Those, and then these circles, these are also made of membrane, okay? So we have lots of membrane inside of our cells, and that is the definition of a eukaryotic cell, is that it has membrane-bound compartments. There's units inside the cell that are bound by membrane. The membrane creates these little compartments. And so what happens is that you can actually create local microenvironments. You can have very specialized environments within the larger cell. So this is a mitochondria, and this has certain chemical reactions happening in it that do not happen in the ER, which is what this is, the rough ER. Does not happen in the nucleus, doesn't happen in the Golgi. They all have their special functions inside the cell, so we can create these local microenvironments using the membranes. Number two is that we have a membrane-bound nucleus or true nucleus. So here, the DNA that we have inside of a cell is bound inside of a membrane nucleus. The other cell type that we'll look at the end of this chapter, which is called a prokaryotic cell, does not have any of this stuff, okay? There's no membrane inside. It only has this outer membrane. Number three, because it has these divided interiors, it is more complicated, right? It's a complex structure because there's different levels and different specialties inside of that cell. Number four, generally larger than prokaryotic cells. And larger, I mean like 100 to 1,000 times larger, so much larger than a prokaryotic cell, okay? So that's your four defining elements of a eukaryotic cell. So we're going to go through each one at the moment. So number one, let's look at the plasma membrane / cell membrane, which is this gold yellow structure. So this is the outermost layer of the cell. It consists mainly of our phospholipid bilayer. And this is just looking at the phospholipid again. So this is review. Remember that phospholipids has a hydrophilic head. It has a charged phosphate group that makes it polar, which means it's hydrophilic. And then it has hydrophobic tails, right? The two fatty acids are nonpolar, so they don't like to go bind to water. So how do they behave in water? Remember this, right? This is our bilayer. So all of our hydrophilic heads, you can see in gray, all of our hydrophilic heads are on the outside, and all of our hydrophobic tails are facing inside. So there's water in, so if this created a circle, there's water inside the cell, there's water outside the cell, but inside the membrane, there's no water. It's just this fatty acid, okay? So you can think about the membranes as being a very thin layer of fat. Okay. So I want to get to letter A, the fluid mosaic model. So I just want to show you the fluidity. So this is a nice GIF of the membrane, and you can see that these phospholipids tend to organize themselves away from water. So if you disrupt the membrane, they can go back to normal. And the fluid mosaic model refers to, so this fluid mosaic model, this is a GIF of, it's trying to illustrate the fluid nature of the membrane. So fluid refers to the fact that all these molecules are in motion, that they're kind of moving past one another. You can see these objects moving within the membrane. Those represent proteins. And the heads of the phospholipids are moving around. So these guys, they're not fixed in place, right? Everything's kind of moving around. The mosaic word refers to the fact that the membrane is made out of many parts. It's a mosaic. So when you look at a membrane, the membrane's made mostly of this phospholipid bilayer, but also proteins and glycoproteins and different things in it. All right, so I have that in for you in the notes. The membrane is fluid, in constant motion, as well as a mosaic and made of different molecules. So let's look at those different molecules. So number one, we already looked at, right, the bilayer of phospholipids. Number two is cholesterol. So I'm moving to, this is another image of, from your textbook of the membrane. You can see the purple actually represent proteins. So proteins are the biggest part that make up your membrane. But let's look at cholesterol. And what I'm going to do is I'm going to just use the picture that you guys have in the outline. So this is what you guys have. So we are going to label the different parts of this membrane. So let's take a look at the number one, bilayer phospholipids. Okay, and I'm going to go through this quickly, but just pause and label your image when you need to. So let's see, what letter is going to refer to a phospholipid? If you look around, what letter is going to look at a phospholipid? It's letter A, right? So A is a phospholipid. And then A is broken into two subparts, so A1 and A2. So what's A1 going to be called? That should be the head, right? And we can go further and say it's a phosphate head. And we can go even further and say it's a hydrophilic phosphate head. And then A2 refers to the fatty acid tails, right? And those are hydrophobic fatty acid tails. All right, so that's A. B, all right, now that we're getting into, actually, let's look at cholesterol too. So cholesterol is number two on your outline. Remember what cholesterol looked like? It was a 4-fused ring structure. So what in this picture could be cholesterol? It's letter E, right? So cholesterol is E. You can see the 4-fused ring structure here, and then notice how there's a cholesterol molecule here, there's a cholesterol molecule here, there's a cholesterol here. So it's embedded in the membrane. So what's cholesterol doing there? It maintains the constant fluidity of the membrane, so the molecules don't move too fast or too slow. Okay, so that's important for the cell. You can't have the molecules moving too fast or stuck in one place. Number three, proteins. Proteins are the biggest guys in this picture, so let's take a look at some proteins. The proteins would be G, or F, or C, and even this guy here that's hanging on one side, that's a protein as well. So there are different kinds of proteins. They have all different kinds of jobs. In your outline, you're going to see the word peripheral protein. Peripheral proteins are just on one side of the membrane. So what's going to represent our peripheral protein is going to be this guy right here. It does not have a letter, right? So you just have to draw a line to it and call that the peripheral protein. And then you have other proteins that go all the way through the membrane. So those are called integral proteins. And so this one, letter G, can be an integral protein. Actually, you know what? Letter F is a peripheral protein too. So letter F, because it's only found on one side of the membrane. Letter C can be considered a peripheral protein. However, letter C has a thing stuck to it. What is this? Remember? So do you guys remember what is pictured by little hexagons? Those are sugar groups. So the sugar groups that are linked up together, how would you classify this? Would this be a monosaccharide, disaccharide, oligosaccharide, or polysaccharide? So the answer is, oligosaccharide. It's not a polysaccharide. Polysaccharides would be much, much bigger. But there are a few sugar molecules hooked up together, and they're attached to the phospholipid here, or they can be attached to the protein. So let's look at number 4, carbohydrates. Usually attached to a protein or a lipid for identification. So remember I told you that oligosaccharides are meant to tell other cells what that one cell is, right? It's a marker of what kind of cell it is. So if it's attached, so you want to use the word glyco, the prefix glyco is for sugar. And so the word glycoprotein is this guy. So this letter C is a glycoprotein. It's a protein, but it also has a sugar group chemically attached to it. So it's not just a protein, it's a glycoprotein. It's a sugar group protein. How about this guy, letter B? This one is a glycolipid, right? Because a phospholipid is a lipid. It has a sugar group, or this oligosaccharide, chemically attached to it. So it is an oligolipid. I'm sorry, not oligolipid, glycolipid. All right. So those are all the parts of our picture here. The H and I, all right, so some people like to ask what does H and I represent? That's just the phospholipid bilayer, all right? So this is one, H is one side of the phospholipid bilayer and I is the other side. All right, so what does the membrane do? What's the function of our membrane? The function of our membrane is to be a selectively permeable barrier. It separates the inside from outside of the cell, but it decides what can go through it. So this is really important. I'll just read what's in the notes. This means some molecules can slip through or permeate through the membrane without being blocked. Others cannot pass through without a channel protein to help it. So a channel protein would be like letter G. It's a tube that runs through the phospholipid bilayer, and if a molecule is allowed to go through that tube, then it will go through it, right? So that's a channel protein. And so that membrane either has the channel protein for a certain molecule or it does not, and different cells have different molecules that come in and out of it. So the membrane is what really stops or allows different things to come into the cell. Okay. All right. The next topic here, letter C and letter D, is the extracellular matrix and junctions. The word extracellular matrix just means what is outside the cell. So if you look at this picture here, notice we have this phospholipid bilayer, right? All the green guys here, those are proteins that go all the way through, so we can call those integral proteins. And then what is outside? All this junk here that looks like feathers and lines and worms, but it's basically protein fibers. So the cells that you have in your body are going to be anchored to things outside of it. And that stuff on the outside is the extracellular matrix. Okay, so just know the definition. It's what's outside the cell. What are junctions? Cell junctions. So cells have to attach to each other, such as a tissue, like your skin, right? Your skin, all the cells of your skin have to attach to each other so that they don't just come off your body, right? They're anchored to each other, and they're also anchored to the tissue beneath it. So, if you think about when you get a sunburn, your skin usually comes off in sheets when you're peeling, right? And that's because all the cells are attached together with these junctions. So you can kind of see the pink here is a junction. This is a different kind. There's different kinds of junctions, but I just want you to know that junctions exist and what they are. All right, so moving on to number 2. The cytoplasm. So I don't have a separate picture for the cytoplasm. Let's just go look at our big cell here at the very first. The cytoplasm is this teal background. It's what all the organelles are embedded in. Think of the cell as full of jelly, okay? So this is very thick and viscous. So even though it's water-based, just like jelly, there's water in jelly, but it's also very thick because there's lots of molecules dissolved in it. Okay, so the cytoplasm is the internal environment of the cell. It's watery, very thick, and it will suspend organelles and nutrients and all these different chemical reactions here. So the cytoplasm, also called cytosol, is very important for the internal environment of the cell. Number 3, the nucleus. So let's look at the nucleus page here. Oops, I passed it. So here's the nucleus, right? This is our circular structure. This is the biggest organelle, and it's something that you can see with your eyes when you look at a cell. And it contains the DNA of your body inside of any living thing. The DNA is inside the nucleus. Well, I shouldn't say any living thing, but for eukaryotic cells, the DNA is inside the nucleus. It's inside in the form of chromatin and chromosomes. So I know the picture says chromatin here. We'll talk about the difference between chromosomes and chromatin, but DNA is the little light strings. And then what is this darker thing here in the middle? That's called the nucleolus. So if you look at your notes, letter B, the nucleolus is the darker staining structure inside the nucleus. It creates or synthesizes rRNA, little r, RNA. This is a building block of an organelle called ribosomes. That little r stands for ribosomes. So we have kind of a different little organelle inside of our nucleus that churns out ribosomal RNA. Okay, now the nucleus, if you take a look at it carefully, we have an envelope. Actually, go ahead and add this to your notes. I failed to make that edit. The nuclear envelope is what you call the membrane that surrounds your DNA. And the nuclear envelope has nuclear pores. So you have holes in the nucleus because there are things that need to come in and out. There's molecules that need to talk to your DNA and access your DNA, and then structures from the nucleus need to leave. So these pores are openings for those. And you can see the nucleus from the outside, the surface of the nucleus with all those holes. Those are the pores. And then this is just a close-up of those pores. Okay, so let's move on. So we have the nucleus. Then we have our next organelle called the ribosome, number 4. So ribosomes are just this blue guy. So there are two, they look like rocks or pebbles, but they're called subunits. There are 2 subunits of protein that create the ribosome. Okay, so in your notes, 2 small protein subunits. You can see here's one, here's another one down here. And the function of ribosomes is to create or synthesize proteins. So I have protein synthesis. Ribosomes, when we look at how DNA is used and the job of DNA, we're going to talk about ribosomes. Ribosomes will come together with messenger RNA. The little M there is for messenger RNA, which is represented by this pink guy, and then it just makes protein. So this is your protein. Remember how in chapter 3 we talked about proteins and how proteins are chains of amino acids? Well, this is each little ball here represents 1 amino acid. So this is the protein being made. Okay, so that's ribosomes. Okay, so ribosome itself is just the blue, just 2 subunits of protein on top of each other. Number 5, the endomembrane system. So this is interesting. So inside of a cell, there are organelles that share membranes with each other. And those organelles are listed for you here. The nucleus, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, and the vesicles. And it's also in your outline, okay? So what that means is if you look at this flow chart, here's our nucleus. The yellow is the nuclear envelope. Notice how the nuclear envelope just kind of comes out and then becomes the rest of this stuff, which is the ER. So the nucleus and the ER basically have that same membrane. It's a continuation of the same membrane. And then look what happens here. The ER will form a little bubble. This will pop off, bud off. Actually, let's go here with the arrows, right? Here's a little bubble being made, pops off. At this point, this is called a vesicle, okay, the word vesicle. And the vesicles will come and fuse with the Golgi. So the Golgi apparatus here is going to take on the membrane from this guy. So they're sharing a membrane. And then the Golgi will create something called a lysosome. And then these vesicles will fuse with the plasma membrane at the top. So in fact, I have the word plasma membrane in your notes, in your outline, but I forgot to put it here. So I should go ahead and add that here. Okay, so magic, I did it. All right, so let's move on to, so that's the endomembrane system in a nutshell. So all the membranes are continuous with each other in some way. They share the membrane. Let's look at each one in particular. So let's look at ER. So number 6 is the endoplasmic reticulum, abbreviated ER because this is a very long word to say over and over again. This is a network of connected tubules and flattened sacs. It's connected to the nuclear envelope. And there's 2 kinds. There's rough ER and smooth ER. Look at your outline. What is rough ER? Rough ER is studded with ribosomes. So you see this picture here where you have these light blue dots everywhere. That light blue dot is a ribosome. So their ribosomes are tiny and they are all on this ER membrane, which is why this is called rough ER, because if you took a picture of it, it would look rough because of ribosomes. What does rough ER do? Rough ER makes proteins that are going to be secreted from the cell. So ribosomes make proteins. Ribosomes are here, so we know that we have some proteins being made. But if it's made in the ER, it's going to be transported out of the cell. Ribosomes can also exist in the cytoplasm. So ribosomes will be around the cytoplasm, and then if the proteins are made there, usually the proteins just stay inside the cell. What is smooth ER? So over here, we have a little bit of ER that's smooth. There's no ribosomes. So what's the job of smooth ER? It's to detoxify the cell and to make lipids like cholesterol and phospholipids. So this guy makes the lipid parts. This guy makes protein, okay? And also detoxify. So if this is an organelle that's job is to detoxify, then what organ in your body do you think has a lot of smooth ER? And a lot of people say liver, and that's right. The liver, which is responsible for detoxifying a lot of things in our body, has a lot of smooth ER inside of its cells. All right, let's move on to Golgi. Oh, I'm sorry, we have transport vesicles. So jumping to number 8. 8 is a vesicle, small membranous sac that carries substances around in the cell. So notice that this is the ER, right, the rough ER. Here's a ribosome stuck to it. We're spitting out a new protein. Here's the protein. So, but the protein needs to go somewhere. So it's going to get packed in this little balloon part. It's going to pinch off, right? And then when it pinches off, we call this a transport vesicle. So it's going to move the protein product, right, somewhere in the cell. So that's what a vesicle is. Okay, here's Golgi. So Golgi complex, or also known as Golgi apparatus, it's the same thing. It's a series of flattened sacs, kind of like pita bread. So if you think about pita bread, how it's hollow, this is like 4 to 5 or anywhere from 3 to 5 pita breads stacked together. And they help to, right, the function is to modify substances that come in from the ER. So what does modify mean? Modify means to change slightly. So maybe a protein coming from the rough ER needs an oligosaccharide. We need to modify that protein to add a sugar group. That would happen at the Golgi. Okay, so the Golgi apparatus receives from one end. So it has a receiving side, and it also has an exit side. So it has an in and out. So here's the transport vesicle from the ER. It's carrying some protein. This vesicle is going to fuse with the Golgi because this is membrane. The purple here is membrane. The red here is membrane. It's the same stuff, right? It's like a bubble fusing with another bubble. So the protein, as it fuses, will come into the Golgi apparatus, and the Golgi apparatus will modify it, packet it up, and then when it's ready to leave the Golgi, another vesicle will form carrying the product, and then it can maybe fuse with the plasma membrane, and then it's out of the cell. All right. So those are vesicles in the Golgi. Let's look at number 9, the lysosomes. Oh, question before I move on. So go ahead and pause it, think of the answer. So what is not part of the endomembrane system? It's the mitochondria. We haven't gotten there yet. So maybe that was a really big clue as to what is not part of the endomembrane system. All right, so this is a lysosome. A lysosome is a small sac with digestive enzymes and an acidic interior. So it's kind of like the stomach of this cell. So when you have, there's 2 ways that lysosomes are used to digest molecules. So let's say this is one of those amoebas or those ciliates that we saw. It's going to eat. So it takes a little bit of substances from the outside. The membrane will form a little pocket and pinch off. So there is our vesicle with our food in it. This is the lysosome. So those dots are going to represent enzymes and acid. And then the lysosome will come fuse with this food vacuole. And then when they fuse, those enzymes inside will start to work on and degrade and eat up that food into littler parts so the cell can use it. So it's like a digesting function. Other ways the lysosome can be used is to recycle parts that aren't functioning anymore, you don't need. So just like your own house, if it's a piece of furniture you don't want anymore, it's broken, you want to recycle it, you're going to break it down and make something else out of it. That's what's happening here. So the lysosome has enzymes This is a mitochondria. It's wrapped up inside of a vesicle. You dump the enzymes in and you break down the mitochondria so you can use the parts for something else, okay? So the lysosome is a digesting little tiny sac. It goes and it can fuse with a vesicle that needs to get broken, with substances that need to get broken down. Okay, which type of cell would probably be the best to study lysosomes? Take a look at what you think. The answer is letter C, immune cells. So if lysosomes are degradative, if they break things down, out of these cells, immune cells would be the most active in breaking things down because they're supposed to look for bacteria in your body, look for dead cells, and then break those things down. So that's your answer. Okay, let's look at the word vacuole. A vacuole is usually a really big space inside of a cell, and it can be used for a couple things, but I'm going to just look at two specific kinds of vacuoles. Number one is called the central vacuole, which is pictured here. And this is just simply where plants will store their water. And you can see how big a central vacuole is. Most of the cell is actually the central vacuole. If you're a plant, water is very important to you, and you keep it in a very big central space. If you are a paramecium, this example, you will have a space inside of your cell that will fill with water, and then you squeeze the water out, because what happens to you in water is that the water constantly comes into your body. It just flows in. If you don't get rid of it, you'll explode. So this is called a contractile vacuole that can pump the water out. Okay, so these are two examples of a vacuole. All right, our last one, well, not our last one, but number 11, the mitochondria. The mitochondria is a really important organelle. It has lots of features to it that you're going to need to know very well for the next unit. So let's take a look at the parts. The mitochondria has, if you look at your outline, an inner and outer membrane. So let's look at the drawing. The outer membrane is obvious. It's just the very outside. Inside, we have another membrane called the inner membrane. The inner membrane is a really big membrane. And so in order to fit this membrane inside of the outer one, you have to fold it up. Okay, so it has all these folds to it. Those folds, you don't say fold, you say cristae. Okay, so you would say the inner membrane has cristae. Then you should know that there is the centralmost part called the matrix. So the arrow should be pointing in here. So in this middle part here is a better place to point. That would be the matrix. Okay. So the interesting thing about mitochondria is that it has its own DNA. It's called mitochondrial DNA. And this is going to be something that we'll talk about with the endosymbiotic theory and how mitochondria and chloroplasts were created. But it comes up a little bit later in the year, but the DNA here is very, very important, that mitochondria have their own DNA. So I have a space for you to draw the mitochondria and label its parts. This is a really good drawing to just copy. And then what's the function of mitochondria? It's to make energy. The energy that your cells use is ATP. So just remember that. It makes ATP for the cell. It makes energy for the cell. All right. All right. The next topic, number 12, is the cytoskeleton. So the cytoskeleton is kind of like the skeleton you have in your body. It's a structural framework that holds the shape of the cell, but it's not bone, obviously. It's going to be tiny protein fibers, and these protein fibers are dynamic. They can actually change their length. They can get longer and shorter. They can move. So the cytoskeleton is really, it's not just stuck in one place, okay? So there are three kinds of cytoskeletal fibers. They are called microtubules. So you can see the word microtubule is here and the tube that it's pointing to. There are things called microfilaments that you can see here, and you can see intermediate filaments are pictured here. So intermediate filaments are like between the size of microtubules and microfilaments, which is why they're called intermediate, and micro is the smallest, which is why they're called microfilaments. But they do a couple things. So yes, they maintain the shape of the cell, but they also act as a railroad. So these little dots here, if you wanted to move something from one part of the cell to another, it doesn't float. It moves along a track set up by the cytoskeleton. It also anchors organelles. So you can see the endoplasmic reticulum here. It's stuck to this microtubule, and these vesicles also move along the microtubule. Um, so I mentioned three things, right? The cytoskeleton acts like a framework. It allows the cell to move and to change shape. So your muscle cells, for example, when your muscles contract, your muscle cells get smaller. That possible shrinking of your muscle cells is because of the cytoskeleton inside. And it provides a scaffolding for organelles and provides a railroad for movement of vesicles and moves things around the cell. Okay, let's take a look at these pictures. So this is a microtubule inside the cell in the cytoskeleton. So the yellow is the microtubules. The bluish purple is the microfilament. So we're coloring two kinds of fibers in the cell, but you can see it's all around. And if you look at cilia and flagella, so we're going to just skip number 13 for just a second here and look at cilia and flagella, number 14. These are all made of cytoskeleton. So cilia and flagella, let's look at cilia. This is the short little hair-like projections on the protist that we saw at the very beginning of this chapter. It functions to sweep substances across the cell or to help the cell swim. So swimming is pretty obvious, right? If these little cilia, they all move, they can help propel the guy through water. The letter number 1 in your outline, to sweep substances across the cell. So in your respiratory tract, and this is really good because of coronavirus right now, when you breathe something into your lungs, before it gets down into the very depths and dark parts, the deepest part of your lungs, the airway, the air tract, your trachea, the larger tubes of your lungs, they're filled with cells with cilia. So the cilia has mucus on it, and when you inhale, the dust and the particles that are inside that air rest on that mucus, and the mucus gets swept up towards your mouth. So when you take an inhale, all the heavy junk that you don't want in your lungs gets trapped in the mucus, and the mucus goes up towards the back of your throat because the cilia will push it. It'll just wave and wave and push, and it'll push that mucus to the top of your throat, and then you can swallow it. So it's this protective mechanism. So that's cilia. So cilia can either be on these unicellular organisms to help them swim, or for us, they help line our respiratory tract and move mucus up. The other thing here is a flagellum. So flagellum is the singular. We're used to saying with the flagella, which is plural. It's one very long structure on top of a cell that helps the cell swim. So the classic example is a sperm cell that helps the sperm swim from one area to another. It also helps other things swim. There are protists, single-celled organisms that swim using flagella. There's bacteria that swim using flagella. Okay, so that's a flagellum. Now let's look at a centriole pair. Okay, so this is number 13 on your outline. This falls under cytoskeleton too, so let's find it. This is our centrioles. It's not super clear here, but there's two. They kind of look like those licorice candies, like those really short, sort of hollow licorice candies. Anyways, but there's two of them, and they're at like right angles to each other. We're going to talk more about these in mitosis and meiosis. They help move the chromosomes around, but they have microtubules growing out of them. So their job is to move chromosomes around, and they're made of microtubules. So it's related to the cytoskeleton. All right, so let's take a look at the cell wall. I think we are moving on now. Yeah, so to the cell wall and to plant cells. So I'm going to go ahead and pause it so we can take a rest.